All right, so I'll do a really quick introduction of these great folks. Eryn is with Tritech and is a member of the Western Regional Certified Crop Advisors. Vivian, sitting next to her, Vivian Wauters, is the primary author of the Cover Crop Guide. She took a ton of research that we have funded, a lot of grower interviews and practical experience from UC Cooperative Extension, from CCAs and PCAs, and really distilled that into that. So we're very excited with that product and very excited to have her here today. And then Daniel, who is a grower here in this area. He's actually a walnut grower, but he has an A+ student when it comes to understanding and investing in the biology of the soil. And so he'll share some of his practical experiences and what he's tried on his ranch. So with that, I really want to just go back to something Bill Brush said in the video that I thought was really clever, which is that the microbes in our soil get the first bite at the buffet table, and the way they, what they choose ultimately sets the rest of the table for what the almond tree gets out of those nutrients that are in the soil. So it's kind of a neat way to think about the importance of all of this. Maybe starting with you, Vivian, if you could give us a little bit about the basics when we talk about soil biology. What does that mean? What are some of the living things in the soil, and why does it actually matter? I'll give you this fun microphone, and just make sure you hold it pretty close.
All right. This is, super cool, double mic. So this is kind of fun getting to follow up on the soil biology, and we're going to be preventing you from eating in a little bit, so we'll be quick and talk through some of these things that have kind of been covered in various ways, right? Like Bill mentioned this morning, there's all these living things in the soil, and somebody mentioned earlier about this is the area where we know the least, where we're still really building our knowledge about things. So we may be touching on things that you've already heard a little bit, and reinforcing some of that and building on some of that a little bit. So the biological component of the soil, this piece that gets the first bite of whatever we're putting in, really can span from things that we can see, right? So earthworms, arthropods, all the way down to protozoa and bacteria. And there's a whole soil food web, right? There's tons going on. And even though, as somebody pointed out, we have half of the soil is going to be water and air, depending on where you are at field capacity. And then we have most of the rest of the soil is the mineral component. And then there's this tiny sliver, right? 1 to 5% on that pie chart that everybody's seen of soil that is the biological component, but it's millions and millions of living organisms of various sizes and various trophic levels, that some of them eat each other, and then some of them eat plant matter, and some of them only eat the compounds that some other microbe has broken down in order to make it soluble, because bacteria just sort of absorb these nutrients into their biomass and then die really quickly and turn over and become the food for the next round of microbes. So there's this whole living web that goes from things that are shredding the leaf matter or residue that's on the orchard floor or compost, breaking that down and then breaking it down further, and then down to the level of microbes that are then making those nutrients soluble. And again, as somebody had mentioned earlier, the more we go through this microbial cycle and everything that is getting to the tree, from an organic source is going through this microbial cycle, making it more and more available, which then, sort of zooming back out from this very small level of things being broken down, has lots of implications for the rest of the soil, not just the biological component. So that relates to infiltration and soil structure, the physical and chemical characteristics of the soil. There's also the mycorrhizal fungal part of the soil. That's a very important biological component that at its sort of atomized level is very, very small, but can extend out and be a really important sort of larger scale connective tissue, especially in these soils without so much clay that can have chemical associations with the minerals, with the nutrients in the soil, that the fungal mass can actually create some of that aggregation and have implications for the physical and chemical characteristics. So there's, just a lot of different levels to soil biology, and lots going on down there. And maybe either of you, if you want to build on any of that.
Yeah, we've been talking so much today about irrigation and what to do with limited water and poor water supply, and with the biology, that is such an important part of how to build a better water holding capacity and how the roots can access all of that water. And going back to the mycorrhizal development, we've got this incredible opportunity in permanent crops to establish a strong fungal community. With row crops, it's so much more difficult, possibly impossible, to do it because we're tilling so often. But when we have trees in the ground for 25 years, you really can build a hyphal network. Those mycorrhizae can pass through dry soil pores to reach areas of the soil that do still hold moisture, and then bring that back to the tree. So if you really want to take full advantage of the water storage in the soil, the fungi are key to that. And we have this opportunity to get them really well established with the trees. So we should be doing everything we can to get those guys to grow. And the cover crops is a key part of that, because that's the transfer of energy from the sun down into the ground. That carbon, all of that energy source, is what's going to feed that microbial community, going from energy and the carbon from the atmosphere to matter. And so over time, those cover crops, the grasses, that's a carbon pump. We're just bringing biomass down into the soil, and a percentage of that every year will be stabilized if we don't disturb the soil. And over time, you're going to be able to build a community with all of those trophic levels. But we can do it in a more focused way for farming, because we have this specific goal of food production. And we can look at important things that are going to be more problematic in the future, like cost of fertilizer. So if we know that phosphorus is our most expensive macronutrient, then we should be looking at what kinds of organisms, which species are the best at mining phosphorus. Fungi is one. And then also look at how you can manage your fertilizer inputs to not disturb their growth. And so that means doing those split small applications. And then with the water management, we've been talking so much about whether or not the trees benefit from water down at 4 feet, but we should really be looking at how the microbial community can tolerate the soil drying out in the upper 6 inches. So going back to looking down at the ground, there's a lot more we can do to benefit financially and environmentally from those organisms.
Danny, you want to add about this? Because I know you were talking about the exudates and some of the things that are happening with those microbes.
Yes. Root exudates is a portion of what I want to talk about. First of all, does anyone in here know how many different species of mycorrhizal fungi there are? Raise your hand. Somebody put a number out. Okay, that means there's a lot. So, what would it be like? I'm going to switch gears here for just a little bit. But what would it be like if every person in this building had a personal relationship with everybody, and everybody knew exactly what every person in here was good at. How many of you would take advantage of that system? Everybody would take advantage of it. Well, that's mycorrhizal's job. Mycorrhizal fungi is an interplant connection. And by some research done on the East Coast, it's been documented that one plant, through communication to another plant, can mine a nutrient, send it through the hyphal pipeline over to another plant. Bear with me. I know that's a ways out there, but hang on. That's what I was talking about. If every one of us would have a connection with everyone in here, everybody would be many times better off because we could utilize everybody's talents that they were naturally good at. Mycorrhizal fungi can take and transfer nutrition. But how that conversation works, I'm going to put it in human terms because that's all we can think about. But those terms are, a plant goes to another plant and says, I need calcium, let's say. So this plant needs calcium. Can you mine me some calcium? And this other one says, well, I don't know. Only if you shove me enough carbon. So what's that carbon? That's root exudates. So once the one plant satisfies its debt with the other plant of the carbon, then the other plant releases the nutrients back to the other plant that asked for it. Now that's pretty cool. So, and it works that way in a diverse system, even with water. There's been places where under very minimal water, and by the way, before I forget about this, biology lives on thin films of water on the soil colloid. That's where biology lives. A little bit on soil structure. How many of you know what chocolate cake looks like? You know how much pore space there is in chocolate cake? There's a lot. When you go home today and you take a shovel out, how close does your soil resemble chocolate cake? It's pretty squashed together, isn't it? It means there's very little gas exchange. The water is stored on the surface of the soil colloid where the biology lives. Yeah, I don't know. Maybe I should quit and go on.
All right, sounds great. Very good. We're connecting the soil structure to the soil biology to the irrigation, so it all adds up. That's okay. I think I'm a loudspeaker. Can you hear me in the back? All right, see? The old-fashioned way. Oh, for the recording. Well, I was almost getting to the question part of it anyhow. I was just saying connecting the soil structure, the water, and the soil biology all works as a system. So that's a little bit of sort of what is soil biology, and I wanted to turn now to practical things you can do to improve the soil biology. Cover crops is obviously one of those. We've talked a lot, just about every panel has brought those up. It is really striking. It's an easy topic to talk about here in the North State, a little harder topic to talk once you get south. But maybe if you could each talk from your own areas of expertise, either other practices in addition to cover crops, or even some practical things that you've done with cover crops that have helped you optimize that tool, and then what are some of the benefits that you've seen that you can actually, a grower could see agronomically on their farm?
I work in Ventura County, and down there, we don't have a lot of cover cropping going on. I know that there's some experiments in lemons right now, but for the most part, everybody's felt that they can't afford to irrigate a cover crop, and we just don't get the rainfall. So in order to try and grow some of those microorganisms, we apply carbon sources along with the mineral fertilizer. So we'll do liquid organic fertilizers, and those contain 16 or 17% carbon. And with that, you've got amino acids, proteins, enzymes, all of the components that come from microbial digestion when the product is made. And then we have other options as well, like microalgae extracts, seaweed extracts, humic acids, and those different biological inputs each have their own characteristics and benefits, but the point is to try and mimic some of the organic matter that would be occurring in a natural system. As in our intensive farming systems, we're depleting the organic matter every year, and so we want to try and put something back that's the product of all of the interactions between those organisms. We want those metabolites, we want the food sources, we want the organic acids that act as natural chelating and complexing agents to keep the fertilizers that we apply in a plant available form, especially when our soil pH is so high that they're prone to precipitation very quickly, especially in colder months. So if you're not able to do a cover crop, I think that there's a lot of benefit you can get from applying some organic fertilizer or amendment sources along with your fertilizer.
That brings up a good point. There's it's like the other side of not cover cropping that Luke was mentioning, where up here you can grow resident vegetation and that can accomplish a lot of the benefits, or if you can't cover crop for other reasons, you can use these other materials. And it points to how application of these principles is really going to be site specific, right? And growers, like, you are the expert on your piece of ground and can figure out and observe, and it's super cool to see what your land actually needs by doing the soil sampling, by looking at how your irrigation needs to work on your orchard. So like Josette mentioned, I do most of my work on cover cropping. And I'm in a lab at UC Davis where we're looking at diversification more broadly in perennial systems. And one of the most impactful things that we've found is by starting with the soil biology, that the more regular, diverse set of inputs that you're putting into your soil, the more life your soil has. And the very practical implications are increased soil organic matter, increased water infiltration, resistance to extreme climate events, right? Like the atmospheric rivers, or resilience in recovering from those. So what we've looked at in almond orchards in the lab is both cover crops, so winter seeded, terminated in the spring so that you have a clear alleyway for harvest, but then also more permanent vegetation that serves as an understory and creates sort of a thatch in the orchard understory for anyone who's going to off-ground harvest. And there's a lot of excitement at UC Davis for off-ground harvest. We'll see where that goes, but that could open up amazing opportunities to build up soil biology. Integrated in with off-ground harvest and opening up opportunities for the orchard floor is integration of animals. So there are people who are integrating animals back into orchards, into vineyards, almonds, walnuts, all of these different systems, because the manure that animals provide and the targeted disturbance that they provide to the soil, as opposed to tillage and the way that they're eating the grass and the processing that they do in digestion. What our lab has found is that that is so key for, as my PI says, juicing up your soil biology. It just gets really juiced up. And in practical terms, that means that those orchards generally, after long-term management, have higher soil organic matter, higher infiltration rates, higher potential resilience to these extreme climate events, and therefore lower input needs. So these are some of the sort of like more out there practices, again, tailored to your specific orchard, that can result in these pretty dramatic shifts in how we're thinking about the orchard. And then also sort of moving beyond, so we've talked about how looking up isn't so great and we really need to look down, but then going back above ground, that these diversifying strategies of putting more life into the orchard besides just the almond tree, and then the soil is the medium that the almond tree is growing in, that you have potential above ground benefits. So for instance, down south, Houston Wilson is looking at how cover crops can interact with navel orange worm management and how potentially there's some synergies there. There's potential benefits for pollinators, or if you have an orchard that is in a slightly less homogeneous landscape, that we can actually support some of the native insects of the area. And also that can increase the effectiveness of the honeybees that you're bringing in. So that can be a cost savings thing if your honeybees are more effective at doing their job. So there's also the biodiversity and soil biology benefits. The same things that you're doing can potentially help with above ground as well.
Okay, so some practices that I have put forth on my farm. First of all, the reason I got going down this road was because of root lesion nematodes. We planted an orchard into a field that planted trees into a field that we didn't know had root lesion nematodes, which caused a myriad of problems. But so people ask me, what kind of cover crops do you use? And I don't really have a good answer, other than the fact that you're going to have to know your soil, going to have to understand the context in which you're growing in, and then Green Cover Seed out of Bladen, Nebraska has a cover crop calculator, and you can take and enter in, this is a resource that I use, you enter in your three topmost priorities, whether it be nutrient cycling, compaction breaking, grazing, you name it. And you can formulate a cover crop mix on that. And you can take the principle, what that gives you, and you can go to your local seed dealer and buy your seed. You don't have to buy from them, but you can use it as a tool. So I had pretty good success with brassicas to reduce my nematode numbers. Nevertheless, that orchard still looks pretty terrible because it was so damaged. So in terms of water penetration, infiltration, and consumption of water, as mentioned previous, that by raising your organic matter, you can drastically increase. I just did some simple figures back there. But if we were able to raise our organic matter 3% on an 80-acre field, that's 4.8 million gallons of extra water storage. That's 14.7 acre feet. This building if I calculated right, is 125 square and 20 feet deep. One of these buildings full of water up to the eave is 2.34 million gallons, which is 7.16 acre feet. So on that 80-acre ranch, you could store 2 of these buildings and have 2 of these buildings full of water greater as a surge tank in your soil that is always there, that makes your irrigation timing less. I mean, please understand, I'm not advocating that you forget about your irrigation, because it is very important, but it makes it less critical if you turn it on tonight or tomorrow morning. So, how do we increase soil organic matter? We have to have green growing plants. Not only green growing plants that, I'll back up. Plant material is not entirely responsible for organic matter production. What is more important for organic matter production is the sugars that are excreted out of the roots, the waste stream of the plants, which grows biology, which can stabilize your soil carbon. So when you stabilize your soil carbon, you transfer that into organic matter, and also the further breakdown of organic matter is humus. There isn't a soil that I've been on that has had too much humus. In fact, our humus levels have been dropping dramatically. So with that in mind, you cannot, I'm going to make another statement here. Evaporation off the soil surface, having a bare soil surface, is far more detrimental than growing plants there, because nature cycles water very efficiently. That doesn't mean that that those plants don't need water. It just means that in a diverse system, everything works symbiotically together. So on my farm, I have found out that even though I grow a dense cover crop and I have lots of biomass out there, I'm not irrigating as much later on in the summer, because I can infiltrate water better and I can build a better soil profile because I have more pore space. For instance, just the other night, I wasn't able to get around to my sprinklers, and I had 2 sprinklers in fairly close proximity that I should have fixed, but it was dark. I couldn't see, and my pump came on. So they were out there shooting a half-inch stream, a half-inch geyser all night long. The lake that was out there wouldn't fill up this corner. It all went down. I have 0 runoff during the year. In the last 5 years, I don't think in all the seepage water that has come, largely spring of 2017, I live right by the river and have seepage problems. I probably ran an average of 3,500 gallons a minute off of my 200-acre ranch of seepage water a minute every day for 40-plus days. I don't think I lost 100 pounds of soil. The water was clear, and it was kind of like tea because of the organic matter that was decomposing out there. But it was very amazing to me that I didn't lose any soil. I had enough soil structure that I could keep all my soil. Now, back to where the lake was. It was along my driveway, and I have it sterilized from the tree to my driveway. And it was a lake out into that place that didn't have any plants growing on them. It was bare. I figured I would be okay running my four-wheeler across that about 24 hours after I shut the pump off. I was driving where it was standing water with no consequences. I got out into that bare piece where there was nothing growing there, and I left tracks in soup that deep. Folks, it makes a difference, a huge difference. Soil structure cannot be replaced. Anybody, any plant can push a root through soup. But you take the water away, and you have a brick, and you can't push a root through a brick. Not very easily.
Great. Well, one last question from me, and then we'll open it up for a couple of questions from the panel, and then obviously lunch is ready. So I think it's really great to hear the upside of these practices, but there's always a reality that that we have to work with, and a lot of growers like to think about return on investment. And I think we know that some of these practices that to focus on building up the biology of the soil, which returns to your soil structure, your soil chemistry, and what that means for nutrient and water availability of the tree, that some of these take a while to see some of those benefits. I wonder if you could talk about either some of the challenges or that time frame of making that investment before you start to see those benefits.
Yeah, I think that a lot of these benefits that we're seeing, this structure especially, you don't see that the first year of implementation. Sometimes it could take up to 10, I don't know, and maybe it's situational, and sometimes you can see a difference in the very beginning. I don't want to speak for everyone, but I'd say don't get discouraged if you have to wait a while, because it's worth it to invest in that land. The improvements that you make now, you can measure them annually, and that will show your progress and make you feel better about continuing and knowing that you're not doing it for the benefit this year, for the next 5 years. It's really so that the next generation can continue to farm and do it better with fewer resources and with more pressure, because it's just getting more difficult. But we've made a whole lot of progress, and as our measurements get better lab analyses available, genome sequencing of soil, we're going to have better ways to improve the soil biology, and we'll be able to prevent some of the other negative consequences, like planting a cover crop that harbors the nematode that you're trying to get rid of. That's definitely one to watch out for. But yeah, keep going.
So some of the challenges that we've seen and that I've talked to a lot of people about with cover cropping in particular is that some of those changes that you can see the benefits from, they don't last. So with cover cropping alone, you may increase your water infiltration when you have living roots there, but if you're returning to a bare orchard floor for harvest and it's perfect and it's flat and you can get those nuts off it, it's super easy, you probably have lost whatever benefits you had for that spring water infiltration. So you're gonna need to plant it again and again and again. And that can feel, I think, a little bit, I don't know, Sisyphean, like you're just doing the same thing over and over again. But if it's an annual practice and working that into your rotation in order to get those benefits, that's something that is ongoing research about when using something to increase your soil biology reaches a sort of tipping point where you've really changed the system versus something that is helping you potentially just within the season. And so I think that's, again, another observational thing. Academic studies are usually designed on a 1 to 3 or 4 year cycle, which is often just not quite long enough to see some of those long-term effects. So I think that's a detriment on our part as researchers of not looking at that long-term impact. And when we do end up looking at orchards that have had implementation for, say, at least 5 years or at least 7 years, that's usually where we see the big differences from these practices. So there is a long-term horizon for some of these practices, sometimes economically as well. So some a recent publication from some colleagues at UC Davis with Jeff Mitchell and Alyssa DeVicentis and others looked at the economic return of cover crops. And they looked at almonds and also tomatoes and found that for almonds, it was around 7 or 8 years where you get economic returns based on a whole set of assumptions about the cost of the cover crop and management and the time it takes to learn how to do it well, and then what kinds of benefits you might get from potential water savings or soil aggregation, lack of compaction, those kinds of things. So there's definitely a time horizon that you need to be considering, and your trees are a long-term investment. So these practices to build up your soil biology and really be working with the soil as a living ecosystem within which the trees are embedded is an important frame shift from some of the more short-term gains that would be great, but that's not really how soil works.
So one of the negatives that I have encountered with cover cropping, and that is that I have found that there is a healthy relationship between gophers and clover. And I don't entirely know what to do about my gopher problem. But at this point in time, it's been important enough to me to keep planting cover crop or growing cover crop. You know what? You actually don't even have to plant anything. I have natural covers that are 3 feet tall out in my orchard this year. It turned wet last fall, and my seed's still sitting in the shop. So whatever I have is volunteer. It's still a cover crop. Yeah, cheeseweed might be a weed, but why wouldn't I encourage a taproot to go down? So we're at an almond meeting, and people talk about pollination. There's been a question of whether or not they don't want anything farmers don't want anything blooming other than their almond trees out there because they don't want to distract the bees from the trees to the cover crop. Well, what happens if 5 of your neighbors' fields all have bee boxes around them, and you have something flowering out in your field besides your almonds? Where do you think the bees are going to be? You might have 30 times the bees out in your field, but all of them may spend a fraction of the time they originally would have. But you're still going to get a good pollination or a better pollination because your numbers are so much higher. So there is the frosting. These are just principles. They're not something that's definitive or it's not the reductionist model where you do this and get this. It's not that simple. But you can adapt to the principle to your situation. And I am open on what to do for gophers because I do have a problem.
I can speak slightly to the gopher issue. This is one that came up a lot talking to people about cover crops is once you go through most things, it's like, okay, but what about gophers? So I talked with Roger Baldwin. He's at UC Davis, and he looks at human-wildlife interactions, I think is his technical title, but a lot of times it's gophers and squirrels. And one of the biggest issues with gophers and something like cover crops or having vegetation is not just that they particularly like legumes, so they love that nitrogen-rich, just like we like protein. They like protein. Legumes fix nitrogen. They're adding nitrogen to their system, so the gophers really like it. They also like other juicy roots. They don't really like the fibrous, grassy roots as much. That's what they say, at least. not the gophers, the people who work on the gophers. So the issue a lot of times with this vegetation is just that you don't see the mounds. And so one of the key principles that I keep hearing is to deal with your gopher issue before you then terminate your cover crop and they have nowhere to hide and nothing else to eat except to go to your tree. So persistent monitoring, trapping, keeping traps in your truck as well. There's actually a really great episode with Roger Baldwin on the Sac Valley Orchards podcast about how to control gophers and what they might go for and stuff. But it is definitely a persistent downside and issue and something to be aware of and to balance some of those trade-offs. And would love to hear from anyone else who's either found success or found something that they shouldn't do with that issue.
Great. So I think we have time for a couple questions from the audience before lunch. So the question was around tests to better measure the sort of biology and the benefits of the biology in the soil.
Well, I don't have an answer to your question, but I will say this I'm only a farmer, so I don't know what all goes on in a lab, but there are people here that do. I would like to see a measurement of the amount of glomalin in the soil. Glomalin is the glue that's excreted by biology that binds soil particles together. If we could figure if we could find a correlation between that, we could probably give ourselves a rating on where we're at biologically.
So my understanding of the way that, it was the soil protein measurement. So ACE protein is the term that people are doing in academic labs, and the NRCS is looking at this with a suite of other practices for dynamic soil properties. So they're trying to figure out, we know how to measure the micro and macronutrients, we know how to measure percent sand, silt, and clay, we know how to do texture analysis, but what about these dynamic soil properties? So this is very much, as they said earlier in the chemistry piece, that we have a lot of these measurements, but what they actually mean and how to actually follow through with them definitely is still up for debate. So you can measure your soil aggregates, so your soil structure, you can measure soil aggregation, where you take soil and then you sieve it a bunch of times and see what sticks together. That is a lab analysis that will definitely tell you what your aggregation is, but that is going into the lab. You could also measure CO2 in situ, so you can put a cap over the soil and see what's actually being respired from undisturbed soils. There's still the caveat of how that actually then relates, or like, what do you do about it, or what's the next step, the so what kind of aspect of it. And I think that that's still really in development. And so ACE protein, when it was originally developed, they thought of it as a measurement of glomalin, and then they realized that it's actually really measuring soil proteins more broadly, and so maybe it's actually a better measurement of these other processes. And so the academic community looking at soil biology is very much sort of on the cusp of figuring out a bunch of the things about what we should be measuring and how we're measuring it and how it then relates to function in the soil, or practices that could be adopted, or tweaks that people should make. So it's maybe an unsatisfying answer.
Next Soil Summit, we'll have more research.
Yeah, and there are all those soil health metrics, the protein index and phospholipid fatty acid, and so many soil lab things. So the Soil Health Institute is looking at several different of those metrics and gauging how useful they are at predicting overall soil health changes and how that corresponds to to yield quality, erosion control, all of that. And they're trying to unify the academic community so that they can give more practical recommendations on what lab assessments to get. So I'd say follow their work in the next several years to see how that goes. But in the meantime, I think that that the most practical way to do it isn't to focus in on one protein, but to do those field tests like the circle water infiltration thing and soil aggregate stability, where if you've got a soil aggregate, you pour water over it, how quickly is it going to break down and dissolve? And then you can do bulk density as well. And there are resources that you can get from NRCS and other places to explain all of these different in-field tests that you can do to look at it, and those are pretty easy. You can get them done in an afternoon, and it gives you a benchmark to measure each year.
One other thing for soil aggregation, have any of you used the Slakes app? I saw you point at your phone and it reminded me. So there's an app where you take a piece of soil and you put it into a little dish and you take a video of how quickly it decomposes in the water, and that can also tell you something. So not the lab analysis, but a very practical in-field opportunity to do that. So I think more of those kinds of things will come online to your phone soon, and that is only to the benefit of all of us.
Well, I don't know about you, but I'm getting hungry. So let's just say that the panelists, I think, will be here at lunch. So grab one of them if you have some more questions. I'd like to thank our three panelists. This was great.